The study investigated the association between glutathione S transferase (GST) gene polymorphisms (GSTM1, GSTT1, and GSTP1) and the risk of primary open angle glaucoma (POAG) in a Brazilian population. It also examined correlations between these genetic variants and clinical features of POAG, such as intraocular pressure, IOP, and optic nerve damage. Method study design: case control study with 87 POAG patients and 85 healthy controls, matched for age, sex, and ethnicity. Genetic analysis: GSTM1 and GSTT1, multiplex PCR to detect gene deletions, null, genotypes. GSTP1, RS1695, PCR-RFLP to identify the IL-105 vowel polymorphism. Clinical evaluation: IOP measurement, optic nerve assessment, and visual field testing. Optic nerve damage was graded 0 to 6, based on severity, Table 1. Statistical analysis: odds ratios, OR, and 95% confidence intervals, C, for POAG risk. ANOVA and Mann-Whitney tests for clinical correlations. Key findings: genetic risk factors. GSTM1 null genotype was significantly more frequent in POAG patients, 49.4%, versus controls, 31.8%, with a 2.1-fold higher risk of POAG, p equals 0.018*. Combined genotypes further increased risk: GSTM1 null, GSTT1 plus *, OR equals 2.4, p equals 0.016*. GSTM1 null, GSTP1 IL, val or val, val *, OR equals 2.7, p equals 0.033*. GSTT1 null was more common in controls, 28.2% versus 17.2% in POAG, suggesting a potential protective effect, OR equals 0.53, p equals 0.085*. Clinical correlations: GSTM1 null carriers had higher IOP, right eye, p equals 0.009, left eye, p equals 0.035*. More severe optic nerve damage, right eye, p equals 0.006*. Advanced visual field defects, right eye, p equals 0.033*. Ethnic considerations: the study population, Southern Bahia, Brazil, was highly admixed, African, European, and Amerindian ancestry. No significant differences in genotype frequencies by ethnicity were found, p greater than 0.05. Discussion: oxidative stress and POAG. GSTM1 null individuals lack a key enzyme for detoxifying reactive oxygen species, ROS, which may contribute to trabecular meshwork damage and elevated IOP. Findings align with prior studies linking GSTM1 null to oxidative DNA damage in glaucoma patients. Controversies: some studies, e.g., Janssen et al., 2003, found no association, highlighting population-specific variability. Limitations: small sample size, limited statistical power for some analyses. Self-reported ethnicity without molecular confirmation. Conclusions: the GSTM1 null polymorphism is a significant risk factor for POAG in this Brazilian cohort, associated with worse clinical outcomes, higher IOP, optic nerve damage. GSTT1 null may confer protection, but further research is needed. Implications: genetic screening for GSTM1 could help identify high-risk individuals for early intervention.